Hello, everybody, and welcome to the EEGNet Colloquium 2023. My name is Alan Evans, and I am Scientific Director of the Canadian EEGNet Network, and the Canadian Open Neuroscience Platform, or CONP. The CONP, supported by Brain Canada and local partners across Canada, was created to foster the open exchange of various forms of neuroscience data and software. EEGNet, also funded by Brain Canada and partners, was built upon the CONP Foundation with an EEG-centric mandate. I also co-chair with Professor Pedro Valdes-Sosa the Global Brain Consortium, or GBC. As you will hear today, the GBC is an international network that focuses upon EEG-mediated brain research projects in low and middle-income countries. The GBC makes use of organizational, scientific, and technical features for open science data sharing and tool sharing that have been developed by CONP and EEGNet. This colloquium has been set up to provide an update on EEGNet progress and partnership with GBC. It is a pleasure and a privilege to host this event today that reviews the landscape of these related projects. After many months of activity behind the scenes, we can now plan a formal launch later this year of EEGNet as an open collaborative platform for EEG annotation, data sharing, and analytics together in one hub. We are delighted to have three speakers today who will describe various aspects of this progress: Pedro Valdes-Sosa, Jorge Bosch-Bayard, and Christine Rogers. A few housekeeping points to note. A panel discussion will take place after all of our speakers have finished. Thank you for holding your questions till then. Alternatively, you can click the Q&A button at the bottom of your Zoom window to submit a question, and we will answer them at the end. Please use this Q&A button rather than the chat option. I would like to acknowledge the sterling work of Christine Rogers, Irene Katma, and Derek Lowe in setting up the logistics and AV for today's colloquium. Lastly, EEGNet would not be possible without the support of Brain Canada and the Ludmer Centre in partnership with Brock University and Laval's Servo Institute. With these introductory remarks, let me introduce our first speaker of the day. Professor Pedro Valdes-Sosa is co-director of the Global Brain Consortium, vice director of the Cuban Neuroscience Center, director of the joint China-Cuba Lab for Frontiers Research in Translational Neurotechnology at UESTC Chengdu in China. He is an emeritus member of the Cuban Academy of Sciences and an adjunct professor of psychiatry at McGill University. His talk is entitled EEG in a Global Context. Pedro.
Okay, I'm sharing my screen. Can you see my screen? Or not
Yes.
yet?
Yes.
Okay, so I switched the title a bit now to emphasize that we're working towards electrophysiology as a tool for global health. It's the same idea, but a bit more detailed. So basically, I'm going to talk about why we made this decision for the moment to put forward EEG as a tool for global health. And then Bosch will talk about the implementation of many of these ideas. And then Christine, how this is being also substantiated by EEGNet. So basically, what we have to see here, is what is the GBC? It's an organization following the principles and practice of democratization of brain mapping into low and middle income countries. And I would say disadvantaged populations, even in high income countries. Our projects are collaborative neuroscience initiatives to increase access for lower and middle income countries to neurophysiology, data, access, standards, neuroinformatic methods, and infrastructure. And to look for potential applications for disease disorder discovery and or equitable global mental health. This is the steering committee of the Global Brain Consortium. You can see here that there's representation of practically all the continents and several of the large projects. I'll touch upon them briefly. So the GBC has a steering committee and a program executive committee. The one that they're all related to diseases that are very prevalent. There you see that there's COVID, brain development, biomarkers of Parkinson, Alzheimer. And the first one, which is the global EEG norms, is the research into the methods and gathering of data that then will support all the rest of the projects. And in fact, is the one very closely related to EEGNet. We would say that the Global Brain Consortium, this project, is the interface of EEGNet to the international community. Now, the other thing that we have to emphasize is that there's a close relation to WHO. In fact, you see there in 2016, the first meeting of the Global Brain Projects, that's the photograph, with WHO, which led to the creation of the GBC, and within WHO to the creation of a novel brain health unit, which is directed by Tarundua, who's in the forefront there. She was the one that took over from Shekhar Saxena, who's to the right with the papers. And this novel brain health unit has launched a program for all the ministries of health, for all the health, on how to protect the brain over the lifespan. And this is something relatively new because the emphasis was on more somatic diseases or on psychiatry. Now, this brain health unit belongs to the Division of Mental Health, and it, of course, does include addiction and psychiatry, but now a specialized unit for brain health. We'll hear a bit more about what they're doing. And then we've worked closely together with them. And here I want to stress that the reason that this is so important is that the global burden of neurological and neurodevelopmental conditions is very high. 70% of the burden is in low and middle income countries. And neurological conditions are the leading disorder of disability adjusted life years. This is the years lost due to the disability. And they account for about 9 million deaths per year. You have the link there, but this is well studied by the global burden of disease. In fact, it is a problem for all countries. Okay, so how can neuroscience impact? This was something we analyzed at the meeting in 2016 and are continuously analyzing. We have brain science on the one side, we have the global burden of brain disorders on the other, where I include neurodevelopmental, psychiatric, neurological diseases. And we have neurotechnology, which is called, if you look at its aspect of diagnosis and treatment, is translational research to take it to try to solve problems of the global burden of disease. However, we have a problem. Most neurotechnology and most diagnosis methods developed in neurotechnology are for the highest level, the hospital level and specialized hospitals. Therefore, there's a divorce with the global burden of brain disorders. And the GBC has been emphasizing that there has to be a linkage, and we'll speak a bit more about this quickly later, to primary health level, to population burden of disease. And that entails a scheme where there's instruments that can be used at a population level, there are instruments that can be used in programs, and there are instruments or neurotechnologies that are be used for fine diagnosis for calibration of other methods. Now, why our emphasis on electrophysiology for the moment? Remember, we do not ignore other biomarkers nor other multimodal types of neuroimages. And it is that the EEG, and now more recently with wearable MEG, OPM sensors now, one can measure with a fine time resolution the activity, which fundamentally are postsynaptic potentials, therefore very fast in a very inexpensive way. So if we look at a diagram of this sort, we can see that all the neuroimaging modalities shed different light, but that EEG is poised to be able to be used in a cost-effective way and deployed widely in any economic setting. Now, based on this, the EEGNet scientific and Technical and Medical Committee, which is one of the advisory organs of EEGNet, and which Ann Gallagher and I co-chair. We looked at what are the uses of electrophysiology in any situation related to health or to well-being, and it seems to be three-dimensional. It depends on which part of the lifespan you're looking at. It depends on what level, and that relates to the figure I showed before, of attention to the population or to patients. And then you have the different types of impairments that have to be detected. So it's a very broad area, and we're trying to address the work with EEGNet, to facilitate the development of tools for all of these different situations. Okay, so what are some of the initiatives that have to be mentioned at a global scale that of the groups that are from part of the GBC? I'll just pick out a few. One is that led by one of the members of the steering committee, Michael Millam, and they have, from the Child Mind Institute, they have funding from the Stavros Niarchos Foundation and created a global center for the study of neurodevelopment. And this is an important initiative, and it already has links closely to the Canadian Open Neuroscience platform. Therefore, we see a convergence of interest in having common infrastructures. Then we have the Born in Bradford initiative, where nearly 14,000 kids were followed since they were in utero, that is, say, in pregnancy, and have all these samples of genomics, biosamples, cognitive assessment, epigenetics, and now they're studying EEG, and they are part of the scientific committee of EEGNet. Then, the next thing I have to say is that the GBC has reached an agreement with the International Federation of Clinical Neurophysiology. That is to say, we're reaching out to 62 societies in 58 countries, more than 20,000 members, because these are the people that are interpreting electrophysiological data. And if we want to harness the collective knowledge of that group, we thought it best to create, and that's why Paolo Rossini, who's a pioneer in multimodal imaging, but also is one of the people that's doing extensive clinical trials in Europe, especially in Italy, to see if EEG can be used as a gatekeeper to see people that will convert from mild cognitive impairment to Alzheimer's. And he's also a member of our steering committee. What are the things we're doing with this group? Well, one of them, let's see, what are the major impediments to using EEG more globally? Well, I can tell you that one of them is that in order to be able to develop machine learning methods, we have to be able to, let's say, collectively have open annotation of many experts. So EEGNet, in our collaboration of the GBC with the International Federation of Clinical Neuroscience, is working on trying to obtain crowdsourcing of annotation. And here you see that there are many different databases that are being gathered, many different editors, of which the ones in EEGNet are of primary importance. And then the development of measures and pipelines to detect artifacts, because this is one of the major primary problems of EEG. You usually record electrophysiological data, and it is very full of artifacts, and there's no unique method for detecting artifacts or cleaning them out, and this changes the results of the analysis. So we are focusing on this and obtaining an annotated database first of artifacts, so that then we can standardize the rest of the pre-processing. Here, the other aspect where looking at is where we have already had clean data. We are trying to obtain a database for the most used method and the oldest of EEG analysis based on the Fourier transform, which is spectral analysis or Fourier analysis. And here you see the result of one of the first collaborations launched by the GBC that includes 9 countries. And what we did was to develop norms for the EEG cross spectrum. That is to say, for all activation at each frequency, as well as for all functional connectivity as measured by coherence. And this is a validation, but also a completion of the work started in the 90s by my group in Cuba. Here you see that we used the method of federated learning. People did not have to share necessarily the raw data, though many of them did. What they did share was the cross spectrum. We then developed norms for the activity or coherence. Here you see frequency, that is to say the different, this is the alpha rhythm, which is prominent when you have your eyes closed, from age 5 to 100, and this is the intensity. And then we used machine learning methods to detect outliers, and this was a feedback loop. We could even detect when people had put the electrodes in the wrong order. Let's say they had labeled them wrong. All the studies in the different countries that we have, the 9 countries, were mostly in children and adolescents. We're now increasing the sample size at older ages. And something that's very important is that we found that there was a batch effect. That is to say, depending on the site, there's a difference. And accounting for this difference, in other words, harmonization, is essential to increase the diagnostic accuracy downstream when you're doing analysis of different groups. Now, we have to increase this large database. The group EEG Many Labs, who's studying the replicability of influential EEG experiments. It is also a multinational project, and we work closely together with the members of this group. We have 2 servers, one in Montreal, one being completed in Chengdu, so we can have this database either for the raw data when the permission is granted or for the intermediate data. So this will continue during the years to be quite complete. We're now including data from Pakistan, from more data from the US, and so forth. Okay, so let's see how this can be used. First, its application to COVID. Here you see that according to WHO, 764,000 people have confirmed cases of COVID, and many of them have long COVID, which is diagnosed after 3 months. At least 45% of the people that have COVID have at least one unresolved symptom. They call it long COVID. This is a project that we have launched to see how we can subtype COVID. Here you see a letter of support from the Brain Health Unit, Darundua, and here you see me, this was last week, discussing with Nicolicius, who is the officer from the Division of Mental Health on this program for long COVID. So what is the basic idea? These are the symptoms reported in hundreds of thousands of people, and synthetically, many of them either relate to the brain or relate to the body with implications of brain-body, with a very high prevalence. And we have defined this in an article that I'll show you later as COVID-induced brain disorders. Now, this is the article. Alan and Muming Pu and Mitchell Muming Pu is from the Shanghai Institute for Brain Science and Machine Intelligence. Mitchell Valdés-Sosa is the General Director of the Cuban Neuroscience Center. Our hypothesis is that the symptoms of long COVID can be either produced by direct invasion of the brain or by other mechanisms, let's say brain-body interactions, or simply by stress. In fact, there are reports that just people with stress can have many of these symptoms. the conclusion that in one of the articles in one of the Lancet journals is that the definitions of subtypes of long COVID is unclear, subsequently hampering effective treatment and management strategies. So what are we trying to do to see if we can subtype COVID? And this is one of the examples of the use of electrophysiology. Here, this is part of the study that will be carried out in Havana and Shanghai. The Cuban part started with studies of subjects that were participants in the study that they were all hospitalized. This was at the beginning of the epidemic. And then those PCR negative were discharged, those PCR positive were treated until remission. And the study simply follows up what is the difference between these two groups, that is to say, isolation in the hospital stress, isolation in the hospital stress plus COVID, and comparison with the global norms. And basically, this is the study. I won't enter the detail because this is described in the what we call the harmonized multinational QEEG norms. But you can see that there is a clear separation. These are the groups that we created, PCR positive, PCR negative. And here, there's a group that we take from Cuba that is a subgroup that's taken as the normal group, and you can see that the separation is excellent. Using methods for stable selection of classifiers that Bosch has developed. So this is one example where EEG can be a tool that, together with other methods, can serve to address huge public health problems. A second example is neurodevelopment. And here I show a meeting that also occurred last week, which is the 2nd and 3rd of May, where the Brain Health Unit is moving to try to define measures of brain health in early development. Now, our contribution to this meeting is fundamentally through one of the projects for neurodevelopment that we call the GLAD project, the Global Brain Consortium white paper. This is what we distributed in the meeting. You see here the signatories, and Alan and Damien Fair have been very active here. Damien Fair associated with the HABCD, and we exposed all our ideas here. This is Maria Luisa Bringas, who is a child, has worked with child neurodevelopment as a psychologist, a neuropsychologist. And basically, the examples that we put were the following, because some people came to the meeting saying that the solution to measure population's brain health could be MRI, low cost, low field MRI. But what we argued is that this should be another level. And we gave the example where you can, with just electrophysiology, just 2 electrodes in the ground, you can screen newborn babies and create a audiogram. This was one of the examples we put in. It shows the strength of a situation where you can exclude artifacts, because looking at bone conduction is to see if there's fluid in the ear, and therefore that you can, in certain cases, for a certain application, use electrophysiology. And we also argued that, according to our idea, one has to be able, for example, this is what we have done in Cuba for neurodevelopmental disorders. It is to have a hierarchy where you have behavioral tools, let's say, and WHO has been developing harmonized behavioral tools. Then you can use neurocognitive tests and electrophysiology, and if necessary, the neuroimages. So another example we placed at this meeting, and we've said before, is about early protein energy malnutrition. This is a study that has been led by Janina Galler for more than 50 years in Barbados, comparing 2 groups, children that had protein energy malnutrition only in the first year of life. And then they had a control group matched for sex, for socioeconomic status. And these 2 groups were studied in 1978. with quantitative EEG, and then in 2018. And this is, I think, the longest running study in electrophysiology. Here's some maps with methods developed by Jorge Bosch. And what you're seeing here are the differences between average age of 5 and 45. Now, the interesting point is that we then went on to look whether the EEG as a measure as a proxy for brain state was a mediator between exposure to protein energy malnutrition and cognition when you are 45, 50 with signs of mild cognitive impairment. What we found with this mediation analysis was that the early brain state when you were 5 is a mediator, very significant. But once it's taken into consideration, the brain state, let's say 45 years later, does not affect the brain. So it is an early condition, which indicates that if we can subtype and look at the processes here, more effective therapies could be used to counter the effects of cognition, on cognition. So I've given the idea of why we want to use and why we want to make open source, because up to now, EEG has been very fragmented, databases have not been shared, and there has been no common annotation. And EEGNet comes to the rescue in this sense to create the conditions to be able to do this within the Canadian Open Neuroscience platform. I have to say that many of the topics I've spoken on have been addressed with much more detail in the Annual Assembly of the Global Brain Consortium, and we are now curating and making public in YouTube these conferences. They're very interesting conferences I recommend. To finalize, I want to say that at OHBM, there will be a local organizing committee symposium, which is neuroimaging in a globally connected world. And here, those of you that go or have the chance to look at it, we will again address many of these issues. Alan, the infrastructure, Luna will talk about a network in Africa, which extends MRI, and I will summarize some of the results of the GBC. Okay, thank you very much. That is my presentation.
Thank you very much, Pedro. As I said before, we will hold questions until the panel discussion at the end. So we will move on now to the second talk. And our speaker is Jorge Bosch-Bayard. Jorge is a Maria Zambrano Researcher at the Universidad Autonoma de Madrid, Senior Researcher at the Cuban Neuroscience Center and the Montreal Neurological Institute. He's a professor at the Center for Neurobiology of UNAM, Mexico, in methods development for quantitative EEG analysis, for source localization and connectivity and global normative data. Jorge.
Thank you, Alan, for the introduction. Let me share my screen. Do you see my screen now? Okay, thank you very much. Hello, everybody. Thank you for letting me make this presentation here. I'm going to talk about some of the tools that we have been developing for quantitative EEG analysis that are part of the EEGNet methods available. So what I want to say is that the method that I'm going to talk about are part of the multimodal neuroinformatic platform that has been developed under the umbrella of the Cuba, Canada, China axis. This is a collaborative project that started in 2017, but the collaboration between the Cuban Neuroscience Center and the Montreal Neurological Institute started very long time ago, since the early 90s. And the development of quantitative EEG analysis started in the Cuban Neuroscience Center in the 70s, led by Pedro Valdes, who has continuously developing methods for quantitative EEG analysis. And what we have been doing during all this year is to be compiling the methods that have been developed in collaboration with the Montreal Neurological Institute in the C-Brain and LORIS system, which are the neuroinformatic ecosystem of the Montreal, created at the Montreal Neurological Institute for the sharing of data and methods openly available for science. The first method that we developed for quantitative EEG analysis in the early 90s, this is the general description of this method. This is just based on creating a descriptive parameter from the EEG to characterize normal brain and to be able to measure deviation from normality. The way that these methods were developed in the early 80s were transforming the QE, the EEG to the frequency domain by the Fourier transform, calculating the cross-spectral matrices and using the EEG, the log spectra of the EEG to obtain all the characteristics present in the EEG to study the normal and the pathological brain function. I want to just make a small stop here. Why do we use Fourier transform, which is a method that is developed for stationary signals, when we know that EEG is a non-stationary signal? For the EEG, so for the quantitative EEG, we do not submit the full EEG recording to the analysis. Like we select only very small, very short windows or epochs that are about 2 seconds long, because this has been described that the background activity of the EEG can last for up to 20 seconds as a stationary signal. To select these epochs, quasi-stationary for the QEEG and artifact-free, the selection is usually made by trained specialists. And at least 1 minute of these signals we use for this analysis. What we warranty when we use this stationary signal is that when we calculate the Fourier transform, all the statistical properties of the EEG are contained in the cross-spectral matrices. So when we are analyzing the log spectra, we are not losing a statistical property of the EEG. Why do I stop it to say this? Because this is something very basic that everybody knows, because I want just to emphasize the importance of the work that is being done by the EEGNet in creating tools for annotation, efficiently annotate EEGs, to mark artifacts, a work that have been they are doing with in collaboration with the group of EEG Lab, and Christine Rogers is going to talk about this after me. So the other thing that I wanted to note then is that when we do the analysis, the quantitative analysis of the EEG, since we only selected stationary signal, we didn't include the epileptiform activities or other transient activities in the QEEG. Therefore, the quantitative EEG analysis that is made by the experts is very important and is complementary to the QEEG. It's not the same. They do not report the same activity. They do not report the same properties of the EEG, and both of them are important. So another of the important things that we did from the beginning was to create a normative population that we recorded from the Cuban healthy population from 5 to 80 years old, to be able to create regression equations, age-dependent regression equations that allows to evaluate individual EEGs to calculate the T probabilistic measurement that assess the deviation from normality. The work that we did in this sense, we did this in different ways. The first database that we collected was about 1990, and there was a second wave about around the 2000, and a third wave that was about 2005. We have been putting this data also openly free in LORIS, and we have been sharing all this information. This is the third data normative data that Pedro already talked about, that we shared in 2020. The other thing that we did in the statistical in the QEEG analysis was to develop methods for the estimation of the primary caring at the sources, and the extension of the QEEG methodology for the log spectra at the sources. So we extended the QEEG methodology to obtain the log spectra at the sources, and then we use them as descriptive parameters that allowed us also to characterize normal brain and measure deviation from normality at the EEG generators. And with that, we obtained this descriptive surface of maturation of the EEG at different places of the brain, of the cortex. And we created then the probabilistic image for the sources also. So this is the extension of the QEEG to the analysis at the source. This quantitative EEG toolbox, we incorporated to CBrain in 2018, and this is also part of the M&I ecosystem of open science methods. In 2022, we developed a new methodology for quantitative EEG analysis that already Pedro mentioned, in which we included batch harmonization. We created the multinational norms from 5 to 95 years old. We obtained norms for the log spectrum, and we obtained norms for the cross-spectral matrices. I just want to try to make a similarity to show the similarities and differences between the two things to know what we gain with this new model. This is the schema that I already showed about the old QEEG that we developed before, and this is what is similar and different from the new normative normative methodology. So the first thing is that we included norms for the full cross-spectral matrix. It's not so. In the first one, we only have for the log spectra. The second thing is that there was a cross-spectrum regularization included that improved the condition number of the cross-spectral matrix and made the estimation more stable. Then we go to the, for both methods, we obtain the global scale factor, apply the global scale factor. This is a factor that we apply to the we calculate that is accounting for a percent of variability in the EEG that is not related to the electrophysiology, but for the maybe technical problems like calibration, like the thickness of the skull, or differences in individuals. This step is common for the two, but then we get we apply Riemann vectorization. So it's the Riemann vectorization is applied to the cross-spectral matrix, and a kernel regression algorithm is applied to obtain regression equations for normative purposes. So in the case of the QEEG, the QEEGT, we use a polynomial regressions, and we use kernel regression in this one. Both of them create these norms, and they create they are able to create the T-scores that goes for, in the case of the QEEGT, it goes for the scalp and the sources. But in the case of this, of the harmonized QEEG, harmonized multinational QEEG, they go to, they can go to the batch correction to account for differences due to different devices, as Pedro already explained. And this allows to measure the log spectral at the scale, but also the cross spectral matrices at the level of the scalp, which give information about differences in the cross spectral, so in the coherence or the cross correlation between the different electrodes, which is something new in this analysis. So we didn't have any way to assess differences in the relation between the electrodes before. These QEEG derivatives have been already incorporated to the C-brain as two different methods together with the data, the multinational database created. So this is the plugin of the new harmonized QEEG in C-brain. And what I want to show now is what do we gain with this new harmonized QEEG. So was it really necessary to create a new method for quantitative EEG analysis? We try to illustrate this by an analysis of a dyslexia sample that we processed together with Dr. Giuseppe Chiarenza. This is a sample of children that were evaluated using the Boder test. There is a clinical test developed by Elena Boder in the 70s, and this test classified children in dysphonetics, dysargetic, and mixed in 3 groups. And what we wanted to know is whether using the EEG we can obtain a good discriminant equation that allows to classify or discriminate between the 3 groups with a high percentage of accuracy, but using the quantitative EEG analysis. For this purpose, also, we included a control group of healthy children that were taken from a database created by Roy Young and Leslie Pritchett in New York. So this fact that we have example from another device and another place emphasize the importance of using the batch harmonization explained by Peter and included in the harmonized QEEG norms. So what we did then was calculate the age-corrected, the T probabilistic Z-scores for the 2 methods based on the QEEG and the harmonized QEEG. Then we have applied a biomarkers procedure to identify using the 2 sets of parameters and see which is the accuracy of both methods. The biomarker algorithm that we use is a stable and sparse classifier that Pedro already mentioned in his talk that we have developed in our center. This is a statistical methodology that is especially designed for when you have a small sample to try to get the most stable classifier with a robust estimation of the area under the curve. This is, by the way, another tool that we have incorporated to C-brain although this is not in the public version of the CBrain, this is just only in the local version. We need to put in there. So this is the sample. We had these phonetic children. We have 184. We have 18, 14, and 100 children. Then we used the two classifiers, and this is what we obtain when using. This is the classification that we obtain when using the T-log spectra of the QEEG as for the biomarker procedure. So the classification of the control group is quite good, but the three groups are not. They are not almost separated. It's almost at the chance level. Is around 0.5 the area under the curve. When we apply the same T-log score, the T-scores of the hard QEEG, even that we incorporated many new improvements, the situation is very much similar to the old QEEG. The real difference that we obtain is when we incorporated the cross-spectral, the T-cross-spectral information to the classifier. So when instead of only using the log spectra, we use the full cross spectra for the classification purposes, we obtain a very high percent of the classification between the three groups. This is so this is a very good evidence that the main differences between the three groups are not in the log spectra, but in the relation between the different electrodes. This is the classifiers that participated in the classification equation. As you can see, they are all relations between different sites. Only in T6 is the log spectra in T6 in delta participated in the classification process. This is in consonance. This is consistent with the Boller theory that her theory is based that the differences or deficiencies in the dyslexia children are due to networks, cognitive dysfunction in the cognitive network in the brain. And these results are also a corroboration of the Boller clinical model, but using neurophysiological measurement. So this is, I'm going to, so other, I am just very briefly mention other quantitative measurement that have been developed in Chengdu by Pedro's group that we are going to incorporate in the QEEG methods. This is one paper, this is one toolbox that was developed by Usama Rias in Chengdu to assess the quality control of the leak field. So the quality control of the leak field is a very important step to have a good inverse solution. So many times we have bad leak field estimation, and then of course the inverse solution will give bad result. This is a toolbox that have been developed to assess that we have confidence in our leak field. And there is a bunch of tools that are being developed by Arioski, Ares in Chengdu, also with Pedro. He has a processing pipeline, a multimodal processing pipeline, in which he can process. So he has developed tools to pre-process and process structural image T1 and T2, or another line of using only T2, and he is developing also tools for incorporating the big brain, all the histology of the big brain in the pre-processing analysis. He also is incorporating the analysis of the EEG with the other types of neuroimage. Here is the automatic and manual QC. This is the toolbox of the liquid, or one of the tools here is this, the toolbox of the quality control for quality control. And he's also starting to create a multi-layer analysis pipeline based on the BC Vareta toolbox. That is the latest inverse method developed by Peter and his group in Chengdu. That is the only inverse solution method that solves at the same problem, at the same time, the problem of estimating the localization and the connectivity at the level of the sources. So these toolboxes are incorporating different type of different types of model of image, neuroimage. They're incorporating information from the histology of the big brain, and they are combining also with the EEG, electrical information, and this is a toolbox that is the PhD thesis of Arioski that will be very soon incorporated also to the analysis for GBC, for EEGNet, and as in the M&I ecosystem. So this is all. Thank you very much.
Thank you very much, Jorge. As I said before, please keep your questions into the Q&A button at the bottom of your Zoom screen, and we'll get to all of those in the panel discussion at the end. Last speaker today is Christine Rogers, and she's going to talk about the EEGNet Data Analytics Hub. Christine is the EEGNet Lead Project Officer at the McGill Centre for Integrative Neuroscience. That's my lab, Montreal Neurological Institute. She's also involved in neuroinformatics platform collaborations, such as the MNI Open EEG Atlas and the open source tools for open science, the Loris databasing infrastructure, which is part of the ecosystem that supports EEGNet, and the EEG to BIDS wizard. So Christine, over to you.
Thank you very much, Alan. I'll just share my screen. Can everyone see it okay? I will be presenting today, as Alan said, on the online data platform and analytics hub that is EEGNet ahead of our fall launch. EEGNet is designed to produce a platform for scalable EEG standardization, annotation, and collaboration. So today I'll provide just an overview of the grant and its objectives. As well, I'll provide a recap and an update on the data platform and analytics hub development, including progress, some upcoming milestones, how researchers can look forward to getting started with EEGNet through 2023, and a brief demonstration of some of the recent features added for data annotation on the platform using head tags or hierarchical event descriptors. So for an overview, this is a grant funded by Brain Canada, as Alan mentioned, with our partners, without whom we could not be achieving this initiative. And EEGNet is also an extension of CONP, or the Canadian Open Neuroscience Platform, as well as the Global Brain Consortium. And thank you to our funding partners. So as Pedro mentioned, with the Global Brain Consortium, EEGNet fits into the Global EEG Norms Working Group, among the many other projects that the Global Brain Consortium spans. And in this context, it provides support for standardization and development of these norms, as well as tools and platforms. The governance of EEGNet is fairly straightforward, with links to the CONP as well as the Global Brain Consortium for partnerships and collaborations. It is led by a steering committee for strategic oversight, with a scientific clinical advisory board and a technical committee that oversees the working group, which drives forward the implementation, development, and planning for the integration of these standards, tools, and features, and also provides support for the research assets on the platform that researchers are contributing. At the same time, there's an administration and communications working group that supports all levels of EEGNet. So the grand objectives at a glance are to develop and deliver for the community a unified platform for the sharing and analysis of standardized EEG data sets. And this is at both the national and international level, with the outcome of having gold standard, expert annotated open EEG data sets hosted on this platform that are ready for machine learning and clinical applications, especially the early identification of biomarkers, and as Alan mentioned, translatable for global health interests across the world, particularly interfacing with Pedro's collaborations and missions. So the aims and the technical deliverables through which we achieve this, through which we aim to achieve this, are to deliver a data platform on top of which there are open EEG workflows for annotation and curation of data. In addition to this, there is a data sharing component so that researchers can contribute and share and access data sets, and an analytics hub through which tools can be mounted and interoperable with the data platform. For this, we use several softwares that are open source and developed in the context of the McGill Center for Integrative Neuroscience. And these are Loris and CBrain, which serve as the data hub and the analytics hub, respectively. And with interoperability and backend hooks, they are able to connect the data to the analytics tools. So Loris, as the data hub, serves multimodal workflows for collaborative data acquisition, curation, dissemination, and also standardization. And this is a web-facing portal that users will have login to when we have a launch later this year, interfacing as well with CBrain. In addition to these platforms, the Canadian Open Neuroscience Platform is a collaboration that we leverage for other key parts of the EEGNet infrastructure. That includes the NeuroLibre publishing platform, but also the ethics and data governance framework, which CONP is active in supporting. And to cover a few of the recent highlights and the progress that has been achieved recently and milestones, I'll just note that since the grant launch in September, that itself was delayed by the pandemic and some COVID-19 complications. We've also had several delays along the way due to COVID. These can range from data collection to staffing. But in the meantime, we have launched a pilot data platform at eegnet.loris.ca that is still in the alpha or maybe beta phase. It supports to support EDF bids and basic annotations on a sample set contributed by Sarah Stevenson and our collaborators at Brock. And since that initial launch, we have added features including a visualization, support for more formats, I should say enhanced visualization, and workflows to add head tags to events. And this is particularly key with our collaborations with Scott McCaig, Kay Robbins, and integrating efforts from the community. So in addition to this, as Bosch has mentioned, we have mounted analysis tools on the CBrain portal for high-performance computing clusters that includes the QEEG toolkit. There's additional pipelines in development, including the Pi lossless pipeline and the Brainstorm tool contributed by Corentin Labelle, who is part of Philippe Alboy's lab at Laval University and the Cerveau Institute. We're looking forward later this year to the platform launch with annotation workflows, data sharing, and analytics. And with that, I'll move on to detail some of the recent activities. So in addition to the analytics that we've integrated, the enhanced visualization is what I will show later on in this presentation, pertaining to the EEG signal browser in particular. This work has been driven by Jefferson Casimir, Tyler Collins, and Jessica Calagaro. And it also implements support for community-driven standards, including EEG bids derivatives, which are in development, hierarchical event descriptor tags led by Kay Robbins and Scott McCaig, and additional ontologies that add to the head tag set, including the SCORE clinical ontology, with work that's been implemented by Dora Hermes's team in Talatia. We also offer some tools, which if there's time, I'll get into at the end, including the EEG to BIDS open source converter, which works for researchers using any platform. So today I'm also not going to be doing a complete walkthrough of the platform, but I'll also present additional tools at the end if there's time. So that's the overview of where we're at with the EEGNet. I'll also add that in terms of an overview of the metrics, we have data sets that have been made online already in terms of our key pilot data set, and we're certainly looking to increase this as 2023 goes on, since we have several data sets already in the queue. And for tools, we've already mentioned the tools in development as well as in deployment on CBrain and our beta group of testing users. We also have delivered 8 conference talks and webinars and several publications. So in terms of how researchers can get started with EEGNet, the first step is going to be, as we go through 2023, please do let us know about your data sets and tools. Many of you have already filled out our brief online surveys. There's a data intake form and a tool intake form, and these are just so you can give us a brief summary of what research data sets or tools you have available and what you would like to contribute. So these are found on our eegnet.org website, and you can also look at our resources page to see this portal of dataset and tool lists and links. So after that step, if, for example, if you have a data set to share, you'd want to select how to share your data. So this depends upon 2 important things: what can you do with your data, as well as what you'd want to do with your data. On the next slide, I'll go into some details, but briefly, the options include sharing via the EEGNet platform, which will bring with it some of the features I've already mentioned, which are visualization, annotation, and interfacing with the Analytics Hub, among others. And another option we'll look at is sharing via the Canadian Open Neuroscience portal. The third step is just briefly, very simply, to get in touch with us and to prepare your data and then send us your data so that we can facilitate loading it on the EEGNet platform. A note, as always with data sharing, is that authorship is not guaranteed, but citation can be required if you use a license such as CC BY with your data. However, we don't at EEGNet have the mechanisms to enforce it, as is normal for any data sharing platform. So in terms of the key question of how do you want to share your data, this comes down to, of course, what can you do with your data? The fundamental step here is always to check your consent form. Does it cover open data sharing? In what forms? And if you'll need help evaluating this, one resource that users might want, or that researchers might want to consult, is the Canadian Open Neurosciences Ethics Toolkit, which is available online at the address you see there. It'll also be updated with a new version in June, so we look forward to that. Additionally, if you want to examine the question of what you want to do with your data, many researchers, of course, want to put a downloadable deposit of their data online so that other researchers can access it. But as we've all seen from various other platforms, including EEGNet, there are aspects which can be, there are features that can also be used for your research data, such as visualization on an accessible and collaborative platform. Perhaps you'd like your data to be browsable and queryable with other data sets, or available for interactive quality review with collaborators. There's also aspects like granular access control, databasing it with multimodal data sets, and adding a customizable data citation policy. These are all things that may be of interest to researchers contributing their data beyond just a simple deposit of a download somewhere on the internet. So this is what EEGNet offers, since these are all features provided through the LORIS platform and are fundamental to the EEGNet mission. So in a nutshell, to share your data with EEGNet, or in sharing your data with the EEGNet, you'll be also adding to it the capacity to visualize it on the platform with other data sets, to annotate it with hierarchical event descriptor tags, which are fundamental to having it available for machine learning and ready for these applications. You can also enable community annotation on it if you like to invite collaborators to include, to add tags and to add annotations that way. And it'll also be potentially available to analyze with tools via CBrain on high performance clusters, as you've heard from Jorge Bosch-Bayard. The options for the Canadian Open Neuroscience platform, which is another great way to share your data set, includes linking an existing OSF or Zenodo entry to the Canadian Open Neuroscience portal. More information at the URL that you see on your screen there. You can also host it on the CONP server in some cases where it might be warranted. For example, you might need a physical site to store your data at. And there are options also to host it yourself and link it to the network with a technology like DataLad. So these are two options for what you might want to do with data sets that you'd like to have shared, in addition to sort of the usual deposit on OSF or Zenodo. So the third step is, of course, to get in touch and prepare and send us your data. Below, you'll see, of course, our email, and we'll also have EEGNet open hours starting soon, later this summer, in order to facilitate any questions or troubleshooting or tool access that researchers might like to have. We also offer open source converters, such as the EEG to BIDS app, which can be used on any machine and used to anonymize and standardize your data and convert it to the BIDS format, which is for standardizing metadata documentation. We also offer EEG uploader tools, and we have an SFTP server facilitating data transfer for those wondering how data actually arrives at EEGNet. And we can also provide templates to help standardize your metadata and containerize your tools. One example of that is that the boutiques descriptor template is a key piece of containerizing tools for CBrain, the CBrain portal on the high performance computing clusters. For more information on any of these, I realize I'm going over this rather quickly. You can also contact us, and we can provide many links, information, literature. So back to the slide, in terms of the recent work on EEGNet, we have an EEGNet signal browser, which has enhanced visualization added to it recently with annotation on EEG BIDS derivatives with head tags, and adding also annotation for artifacts with the SCORE clinical ontology. And so I'll show that briefly in a moment. Just to give a brief overview of a screenshot of what the platform looks like, this is at a glance the signal browser. I'm going to show a few things just now as I do a demo. So the first thing is that we do have our EEGNet platform up and running in a beta form at eegnet.loris.ca. When we begin to invite more users in towards the platform launch in the fall, this will be updated as well. And currently we have a few features already on it, such as signal review and 3D electrode maps, but we have more features in development that aren't quite there or are in development before launch. When this platform is completed, you'll see things that are more typical of other LoRis instances, such as some graphs on the inside of the platform that will show data that has been added, and we'll have some presentations of some tasks and some highlights here. And then in addition, the work currently in development on the platform includes the ability to browse and annotate signals. So I'll show this now. So if I, for example, select from one of the channels that we have here in the online database and go to the signal viewer, what I can see here is several signals, and with their values displayed as this, I can increase the amplitude, and I can set filters for this data. But I can also zoom in so I can make a little bit more sense of this data. So for example, if I reset the timeline here, I can use the slider to show a particular time interval at a glance, and it just takes a moment to re-render. There we go. If I'd like to see all of these signals plotted against each other to look for variance at a particular moment, I can use the stack feature. And what this will do is it'll plot them all on the same axis. I will also zoom in on a particular region. So for example, if I'd like to show something in this segment, I can simply highlight the region and click a button so that it zooms in on that region. I'll also switch at this point to a view of 32 channels, which just takes a moment to render. And at the same time, I can look at all these channels and I can move forward through time as well as backwards. And I can stack these as well. And it just takes a moment to render, but you can see that the signals show overlaid on each other. And so you can get a sense of what the variation is in the signal at a given moment in time. On top of this, we can move through the channels and find ones that are perhaps showing greater variance, and as well show events that are also overlaid on this. So it just takes a moment to render. And at this time, let me see if I have this loaded already. You can see here that at this point in time, that you'll see a channel that might display greater variance than others. And for events like this, you can also view the events on the plot and add tags per the head score ontology. For example, I might want to indicate that this tag, this event in particular, is an artifact, for example, where we have this one signal that is demonstrating some variance. So in addition to this, and I'll just go back to the other plot now, which is the same data. You can see here the variance in the signal, and this will show, again, some more events at the same point in time. So in addition to tagging specific events with head and score tags, there's also a dataset tag viewer where a particular tag, you might want to identify an event that has this checker F5 event by its head tag. Long form head tags can be shown here. And in addition to this, you might also want to attach for example, an additional tag to it, such as that it's a queue for a particular task. So you can add tags and annotate data this way. These features are still in development, but they're part of our partnership with the HEAD group led by Kay Robbins and Scott McCaig. And a lot of this work has also been implemented in data sets using the score ontology with Tal Attia and Dora Hermes's group at the Mayo Clinic. So that's a brief overview of what you can do with the signal browser, going back to how signals are collected, annotated, viewed, and also marked for quality in EEGNet. So we'll keep you apprised on that as we get closer to a fall launch, but that's a brief tour of the signal browser. And I'll just mention as well that this work is built on previous projects, including the MNI Open IEEG Atlas, which was a pilot in Loris in 2018 with Birgit Frauscher and John Gottman, followed up by Birgit Frauscher's IEEG Sleep Atlas for intracranial collection at 10 sites in 6 countries that is currently underway. This is an open atlas that anyone can sign up for an account at if you'd like to access some of the visualization tools and get a feel for how the tagging and the data works there. In addition, Pedro Valdes-Sosa piloted the Cuba Human Brain Mapping Project with Jorge Bosch-Bayard and members of our team, which resulted in a publication in Scientific Data. Given the multimodal nature of this data set, including EEG visualization and MRI and other population, clinical, and behavioral data points. This data is also open in the via the Canadian Open Neuroscience platform, if that is of interest. Additional tools that I'll mention just briefly include a converter tool that we've also launched for the community called the EEG to BIDS Wizard tool. It enables users to standardize their EEG data to BIDS format, initially developed for EDF and now supporting MFF and SET shortly. We believe for the spring release, we'll have new versions of this tool ready. And the idea is that any naive user or user who may be a junior member of a lab can run this tool on any computer, including Windows. There's no command line or technical knowledge needed. And it's all run on open standard libraries, such as the M&E BIDS library and the BIDS validator library that are used by the community. It allows you to enter and validate IDs if you are going to be loading this data into a platform like EEGNet or another LoRa's. And at the end of the day, it also validates your data to make sure that it's BIDS compliant before you try to upload it to a platform. So these tools are in development for additional spring releases, including interaction with the LoRa's API, formats, and presets so that templates can effectively be preloaded in the tool. So future extensions for EEGNet as we go forward towards the launch and beyond include increased interoperability with the Analytics Hub and seamless return of results to the Data Hub on LoRa's from CBrain, additional fine-grained access controls so that researchers can control their own data, and visualization enhancements are ongoing. We hope to be adding in the shorter term additional montages, including enhanced electrode maps, the ability to tag based on a subset of channels, and overlay with anatomical data using the Brain Browser open source tool that we have already integrated into LoRa's. And these are all future extensions that we look forward to adding in EEGNet through the grant and beyond. So with that, I'll just wrap up and say thank you for your time here today at our colloquium. I will pass it over to Alan in one moment to do a Q&A with our other speakers as well. And we do hope you will put some questions in the Q&A for our other speakers as well. But I just wanted to say this has been the briefest almost overview of EEGNet's progress and objectives and our upcoming milestones, how researchers can get started contributing data and tools, and as well with a brief demonstration of some of the work that's in progress by our development team, especially Jefferson Casimir, on data annotation with head tags. And this all brings it back to the objectives of EEGNet, which is to deliver a unified platform so that standardized open EEG data sets can be shared in the national, international research community with the annotation layer of head tags so that they are machine learning ready for translation and clinical applications. And the technical deliverables are the data platform, workflows, data sharing, and analytics. And we look forward to keeping you posted on these developments as we look forward to our fall launch of the platform. So thank you very much to our partners and our teams, especially the EEGNet Working Group and contributing developers at EMSN, including our Director of Technology, Samir Das, who's here today. I'll also mention that for anyone attending the Organization for Human Brain Mapping meeting that's happening in person in Montreal in July, EEGNet will have a booth together with Canadian Open Neuroscience Platform and also the Global Brain Consortium, side by side with some other EMSN-based organizations from our lab at the Neuro here at McGill. But in addition, EEGNet will also have a talk. So we invite you to come and check us out. And thanks for anyone who is interested in spreading the word about the talk that we'll have at OHBM and the presentation there. So thanks very much. Back over to you, Alan.
Thank you very much, Christine. A comprehensive presentation of where we're at with EEGNet. I think at this point now we'll move over to the panel discussion, the Q&A session. Feel free to raise your hand. I'll try and keep track of hands appearing on the screen, but also make use of the Q&A button at the bottom of your screen to post questions. There is a question there from Samir Das, answered by Pedro Valdes-Sosa. You can read that, but essentially there's a larger issue that's related to this question, which is, how do I get access to these tools to analyze my data? What does any person, a researcher across Canada or indeed internationally, have to do to get access to these tools? So maybe Christine start that and Pedro would chime in.
Yeah, absolutely. It's a great question. Thank you, questioner. I'll just say that we look forward to our fall launch for making the EEGNet platform as a whole accessible to researchers. Many of these tools, which Bosch detailed for the harmonized QEEG toolkit, are already available on CBrain, and those can be deployed on your data with a CBrain account. If anyone's not sure how to access that, you can certainly get in touch and we can connect you. The other open source tools that we mentioned, such as LoRa's itself is an open source data platform, and CBrain is an open source portal in terms of its code base, as well as the EEG to BIDS converter. These tools are all open source and can be downloaded as software packages for use in different initiatives, collaborations. And of course, since they're open source, they can all be customized for deployment as you wish. So it depends on the context and the level at which you're using these assets. The other thing I'd mention is that the CONP portal is, of course, already online. I may have neglected to show that, but Patrick Bermudez can field any further questions. And I'll just note briefly that the address for the Canadian Open Neuroscience portal is just simply portal.conp.ca. I hope that covers the tools that many of the tools we mentioned. Please don't hesitate if I've forgotten to.
I think you might need to provide someone with one starting point that they go to, and then they can get advice and guidance on how to get access to all the various solutions and data tools that have been described here.
Absolutely.
A
So
lot of links. Just one person I want to talk to is usually how this goes.
yeah, absolutely. So for that, I'll bring up just my slide about how you can get in touch with us. So for that, we invite you to check out our website, contact us through the form there, or email us at contact@eegnet.org. And we'll also be posting more information about open hours, about the ensemble of tools for EEGNet shortly on our website. So those are all options for just getting in touch.
Pedro, you had anything you'd like to add here?
Okay, I had mute off. Look, I think that there's two issues here, what we've discussed, or three issues, no? There's the common infrastructure, that's the EEGNet. Then there's a series of analytical tools that some of them are in CBrain, specifically the frequency domain analysis that Bosch said, and others are in the pipeline, which are the ones that we're preparing. Well, the classifier that we've been showing in several of the figures, the mediation analysis, and let's say the pipelines, which the integral pipelines that Bosch mentioned at the end, and then also we're going to incorporate, because I didn't mention this, but the whole idea of the multimodal pipeline is a connection to something we haven't mentioned, which is the highball project. So one of the things that we're developing to put into CBrain, and that's the next task, we already published a paper on highly efficient method for analyzing neural masses, and we hope that putting it on CBrain will help us integrate this work by Anis Ladies and our group to allow us to model and perfect using inverse methods many of the methods we have. So regarding methods, some are there, some are in the pipeline, and regarding the data, we have all the multinational normative data, the cross spectra, and is already in LoRa's. It's already available. We will make available all the data through LoRa's and through EEGNet, but there is a particular situation where we have to now for the, let's say for the populations or for the samples that come from different pathologies, we are now discussing in which way they can be incorporated due to all the permits, etc., that are necessary for this type of data. One possibility which seems to be gaining traction, for example, Samir asked about the Barbados data, is to share the cross spectrum, because these are intermediate statistics. Now, having said all this, there's going to be an important part where we really need to work, which is to create tutorials with examples because having the tools in CBrain, having the methods in EEGNet or the CBrain, and having the data in LORIS, it does not mean that it's going to be easy for people to use it. So I think that we should have some worked examples going from gathering the data in a certain population, then comparing it to the norms, doing several statistical analysis, and actually even outputting material that can be put into the into papers. And I think that the conception of the Canadian Open Neuroscience Platform as a full cycle thing is excellent for this. So there remains a lot of work to do, and anybody that's interested, this is all open source. Anybody that's interested in collaborating on all these aspects or adding things, they're very welcome. For example, we're working on microstates. I mean, the stuff we've done is not let's say the sum of everything that can be done in EEG. There's much more to be done. Okay.
Thank you, Pedro. We have a question from Samir, from Patrick Bermudez. Patrick, I could read it out, but if you're there, why don't you speak to your own question?
Sure. Thank you, Alan. This is just a general question about the sorts of things that are perceived as impediments or just things, challenges for getting our data into EEGNet, generally speaking. And Christine, I was just wondering, and anyone else can chime in your conversations with researchers in general, what is the most commonly cited impediment? Does it have to do with just even the initial work of finding and organizing your data to make it presentable to the rest of the world? Does it have more to do with REB permissions typically, or other types of resources down the line?
Thanks, Patrick. That's a great question in terms of the actual impediments. We've done several surveys of the EEGNet community to try to understand where we could best provide services and materials and really aid or give a boost to researchers finding some difficulties. Obviously, the ethics is the first stumbling block that researchers will encounter. And so understanding how to navigate the questions of data sharing is key, which is why we make reference to so often the Canadian Open Neuroscience Ethics Toolkit, which is a great way to help researchers think through some of the issues. And as well, we find that there's sometimes a technical barrier in terms of adopting data standards. And for that, we provide converter tools born sort of out of the need to reduce the time it takes researchers to maybe format their data into a specific standard and validate it too. Since there's nothing more frustrating than contributing your data to a platform and then receiving an automated message saying, there was an error processing your data and you're not really sure what that might mean. So we provide those open source tools such as the EEG to BIDS converter precisely so that some of those barriers can be addressed, reduced, or lowered. But we're certainly looking to hear more from researchers about how we can bridge those gaps to get data sharing really a much more intuitive and easy process for the researchers. Time to annotate data, as you mentioned in your question, is another aspect too. We're hoping to bring, as Pedro sort of addressed, a galvanized community effort towards bringing these research assets out into the community for the benefit of many, many researchers globally to help incentivize some of and reward some of the time spent on the platform in terms of annotating data and contributing data and formatting data and preparing it for contribution too. So those are all things at different levels. I think the technical barriers are reducing over time as people get more familiar with the formats, but we are keeping a close eye on that.
I'd like to add to that, if possible. Okay, so we have managed to gather 1,500 resting state EEGs. And I have to say that first, half of the people could not share the raw data because of ethical, the agreements. So looking at federated ways of sharing data is always something, though we think we can get more and more people, and the problem has been legal, especially with the Europeans. So second problem is that people are hesitant still to share their data after the, but the point of this, I think, can be overcome if, as Christine said, you give them more technical facility and they see they can, they can obtain, for example, one of in any of these collaborative efforts, and all the people that contribute are co-authors, and I think that this is something that stimulates it. On the other hand, just as with fMRI, I think that the major stuff, we have to make a clear distinction between resting state, which is boring but easy, and that's what that's the easiest thing to do. However, as for fMRI, even worse, I would say, for event-related responses, there isn't it's not that the data isn't standardized, it has to be converted. There aren't standard paradigms, and that's something that the EEGNet Scientific Committee has been discussing. There isn't a standard. Everybody is testing a specific hypothesis, so the data sets are very small. Either we find ways of generalizing across these different experiments, which seems to be something that people don't like. Specifically, Stephen Luck says that the components change with any type, and also Scott says that, truly, no? That depending on the task, the components change, so it's difficult to have a common annotation across these data sets. So one thing that has to be worked with is, besides the resting state, have a minimal set of standard experiments, because then we're going to be able to gather larger data sets. If not, what we have is a system that will allow everybody to annotate what they're doing in their own lab. Now, the third aspect, and also Christine was mentioning it, is to mobilize the people that use this in clinical work. And these are not researchers always. These are people that are clinical. And up to now, they work with, let's say, industrial company EEG systems, each with a different format, each which not always go to, let's say, a head annotation system or even a common format. And there, the technical barrier with the people who are doing clinical work is even larger. So with the platform constructed, I think that some simple interfaces have to be rapidly developed and deployed where the clinicians, you see, clinicians have a lot of work. And if they do not have the time for a learning curve, for entering the configuring stuff, so some ready-made things have to be given. We've been thinking and discussed this in EEGNet to have, for example, for artifact annotation, just a simple program that goes up, shows them some EEGs that we've discussed with IFCN, and they have to say if they're artifacts or not blindly. And we've done this before, decades ago in Cuba with experts, but it has to be so easy that the clinicians will take the time to do it and then get positive feedback. So the problems are not only technical or computational or anything, they're sociological. They're how we are going to engage people that are going to be using this for different purposes. And for that, we need to have EEGNet up and running to add the things that I'm saying.
So Pedro, one supporting point that you may not be aware about is in CONP, we're newly creating something called CONP experiments, where you can sort of describe in a standardized way the whatever experiment. Our first use case, I think, was more fMRI related, but it doesn't matter. It could apply easily to EEG. So that's something that's on your radar. And to your first question, also just something else to highlight. I agree that showing people how to do the analysis in a kind of a nice way, like a tutorial, is very beneficial. So something like a Jupyter notebook with everything highlighted, including links to data, I think could be very valuable. There is in beta, version of this called Neurolibre, which I think could be leveraged. So there's a lot of supporting tools. And I must say, since 2014, when I first sat with the electrophysiology standardization group at INCF, things have come a long way in terms of putting all this together. But obviously, there's still little pieces like the ethics and getting data access and things like that. So anyway, just thought I'd add a few extra points.
Good.
I'd like to just continue a little bit this discussion of getting access to data and tools. As has been already intimated, there are regulatory constraints. You can't move data necessarily out of your local environment because of GDPR in Europe, as an example, or your own institutional REB regulations. It's possible, if you want to make data available yourself, you can use DataLad. It doesn't have to be sent to CONP, as was already pointed out by Christine. On the tool side, it is possible to move the analytics to the data rather than the data to the analytics. So all you have to do ultimately is to sign up your institution as a node in CBrain, and you get access to all the CBrain tools running on your data locally. And that potentially gets you out of the problem of beating the REB or GDPR regulations on data transfer.
But Alan, there something I want to ask Samir. The fact of federating those results, are we there? We need an extra step. Let me put an example. I've been discussing with the microstate people. Microstates is essentially a cluster analysis. How are they doing it now? In small samples, they're doing a cluster analysis of EEG topographies, and then they're trying to match the cluster centers. And it's well known that if you take subsamples in the data space and do separate clusterings, you may not necessarily be recovering stuff. Now, the solution to that is federated cluster analysis, federated learning, machine learning. So I think a very interesting thing would be, what are the hooks and things that we need so that people can run CBrain local, there, get out of the trap of the ethical approval, and then share data, share results. And I would say that technically there's sufficient statistics. That is to say, the statistics of the data that substitute the raw data. And then once you can combine those, you can do quality control on those subsamples. So I think this is something that would be very interesting.
Yeah, so I do have several answers to that. Number one, in my opinion, just sharing the data openly is your best bet. I have seen other versions of different kinds of federated sharing, including federated learning. And so with federated sharing, like something like, for example, CoinStack, you don't control the algorithms, and there's a constant back and forth, and a lot of researchers just don't like that. They want to control their algorithms. They want to go through the administration bureaucracy of doing it. That said, it works to a point. Federated learning, where you have these these individual nodes that do something, there's still a lot of back and forth to maintain that. So it works, but with limitations. So if we can get around, GDPR's intention was to punish large corporations that are doing really nefarious things with your data. I don't think they thought about research and how this actually helps the patient. So if we can, find ways to, make data more openly available, I think that's our best bet. In the meantime, you can do things like federated learning or federated distributed data sharing, but those have limitations for sure.
I'm not seeing any hands raised. Don't be shy. While we wait, I do have one question, which is more of a scientific interest than a procedural one, but it concerns Bosch. You talked a little bit about the use of the big brain data set to constrain the forward modeling problem. For those who are not familiar, the big brain is a high resolution cytoarchitectural data set with a very high resolution of 50 microns. 20 microns, sorry, in each dimension. So that gives you the ability to extract features in a forward modeling context, which you couldn't do from MRI. So you can model the 6 layers of the cortex. That data is already available publicly, the 6 layers of the human cortex. And Bosch made the point that can be used to constrain forward modeling, simulation environments. And the question, I guess, is Is that a research enterprise that you and your team are following, or is this something that other people can get access to and make use of that same data to explore their own simulation environment?
And I prefer Pedro to answer this question. So this is the work that is being done by two researchers at Pedro's lab under Pedro's direction, is Deirel and Arioski. They have been working hard, modeling introducing the big brain information in the constraint for electrophysiology. There is already a paper published, and there is a toolbox, but I prefer Pedro to explain all of this.
Okay, Alan, as you know, with just the usual one layer, that is to say, in the middle of the cortex, we already have inverse solutions for all the methods, no? Which project out to the big brain, so we can move it. It's not a difficult task to move from standard atlases to the individual MRI and do inverse solutions. We've presented that at several of our seminars. The new work that's going on is to calculate the lead field for each of the cortical layers, because the first question we have to answer, what is the full width at half max of the different methods for EEG? What is the resolution in fMRI? And you've seen that with Camille, who is collaborating with us on putting in the fMRI, the layer-specific fMRI. There doesn't to see you don't have an infinite resolution. So we're now trying to characterize what is the spatial full width at half max. And so how well can we distinguish layers? That's the one thing. And then the other thing is to see how the neural mass models can inform this whole process of generating EEG and fMRI from neural mass models. And that's the direction we're going. It's research work, but as each unit of work is finishing, it's being put on GitHub. It can be shared, and we're going to put it into CBrain at the very least. What we made, what perhaps might be interesting, and I was thinking of presenting something of this at the highball meeting in Iceland, no? Is the overall picture. So what is the overall programmatic picture? And then to gain traction from interactions with other people. No? We've spoken, Alan, of the work done by Ratislav Benšek, Boris Bernhardt, the people there. I think we really now are at the stage of pushing this forward. So I think this is very important because Fernando Lopez da Silva, who you know is one of the main leaders in electrophysiology, and I discussed many years ago, and in fact, that was what motivated a 1999 paper, which is that EEG analysis should be based on neural mass models. That is to say, instead of looking at arbitrary signal processing parameters, such as spectrum or whatever, to be looking at connectivities and activity in models of the nervous system. So that still is, we thought it was more achievable, let's say, in a less time, but I think that's the overall end program. And that's where it ties in with highball.
For those who might not be familiar with the term highball, this is it's an acronym that stands for the Helmholtz International Big Brain and Analytics Learning Laboratory. It started life as a collaboration between the lab of Katrin Amunds and the Forschungszentrum in Germany, in Jülich, and my own lab, the EMSIN here at McGill. But it is expanded dramatically in the last year and a half with about 400 researchers worldwide. And the general premise of highball is to explore multimodal big data analytics with a focus on integration of cytoarchitectural information, electrophysiology, chemoarchitecture, and normal structural functional imaging. And highball is meeting this year in Iceland, in Reykjavik, and one of the threads of this is infrastructural integration between Europe and North America and indeed globally. That particular aspect of the highball activity is very relevant to EEGNet and GBC.
Yeah, I think so. And we'll look forward, Alan, to more integrations of sort of the EEG pillar of highball as we go forward this year. I think there'll be some interesting news. I think with that, if there's no more questions, we can continue discussing, or we can wrap it up now. Thanks very much for having attended. And Alan, I'll leave it to you for some closing remarks.
Well, as always, there are so many things that I think we'd like to sit around over coffee or over a beer and discuss in much greater detail. But I think you can see that there are a lot of developing shoots for an international infrastructure and international scientific integration of EEG data globally at a level that hasn't been possible before. We have the tools, we have the infrastructure, we have the organization and the scientific community engaged to do this, as you've heard today from our speakers. They've covered a lot of territory, both the methods as well as the visionary perspectives of global integration. And I hope that you've enjoyed these presentations. Please do feel free to follow up with Pedro, myself, with Christine, and with Patrick Bermudez at CONP, and we will most definitely follow up back with you. I should like to point out that there is a presentation at the OHBM, upcoming OHBM conference, that it was focused particularly on global scientific integration, particularly into low and middle income countries, and GBC/EEGNet will be presented there by Pedro and myself and Udana Anazado, who will talk about projects in the African context. With that said, I think we've had a good go today, and we'll call it a halt. I would again like to thank Christine Rogers, Irem Katitas, and Derek Lowe for all of the help in organizing this, and of course the speakers. So thank you very much, everybody. Have a good day.